The battle between CRISPR and viruses is one of the most intriguing showdowns in modern biology. On one hand, you have CRISPR, a groundbreaking technology that allows scientists to edit genes with unprecedented precision. On the other, you have viruses, nature's clever, often insidious invaders that have been infecting living organisms for billions of years. But in this contest, CRISPR isn't just a tool of human innovation, it's also a defense system used by bacteria to fight off viruses. So, in this epic biological battle, who wins, CRISPR or viruses? What is CRISPR, and how does it fight viruses? First, let's break down what CRISPR actually is. CRISPR stands for clustered regularly interspaced short palindromic repeats. It was first discovered in bacteria, where it serves as a form of immune defense. When bacteria are infected by a virus, called a bacteriophage, they remember the virus by incorporating segments of the virus's genetic material into their own DNA, specifically in the form of CRISPR sequences. These sequences act like a biological memory bank, recording the viral invader's genetic code. If the same virus tries to attack the bacterium again, the bacterium can recognize the viral DNA through its CRISPR sequences. The bacteria then use CRISPR-associated proteins, Cas proteins, like Cas9, to cut the viral DNA at specific locations, effectively neutralizing the threat. This ability to remember and cut viral DNA is what makes CRISPR an immune system in bacteria, and what inspired scientists to develop CRISPR technology as a powerful gene editing tool in higher organisms, including humans. The role of viruses in CRISPR's evolution. Interestingly, viruses have played a significant role in the development of CRISPR. Bacteriophages, the viruses that infect bacteria, were the original threat that led to the evolution of CRISPR. By integrating viral DNA into the bacterial genome, bacteria could fight off repeated infections from the same virus. Essentially, viruses were the catalysts for creating the very system, CRISPR, that would later be used to edit genes in a completely new and revolutionary way. This form of adaptive immunity gave bacteria an evolutionary advantage by allowing them to recognize and eliminate viruses that had previously attacked them. But while bacteria were evolving CRISPR to defend themselves, viruses were evolving too, constantly adapting to escape detection and destruction by bacterial immune systems. The constant evolutionary arms race between bacteria and viruses has resulted in some fascinating adaptations. CRISPR versus viruses, the battle in bacteria. Now that we know how CRISPR works in bacteria, let's take a closer look at the ongoing battle between CRISPR and viruses in the microbial world. This is not a one-sided fight. Viruses are highly adaptive, and they have developed ways to counteract CRISPR defenses. One of the ways viruses fight back is by evolving mechanisms to block CRISPR recognition. For example, some viruses can modify their DNA to make it harder for the bacteria to recognize and target them. Others produce proteins that interfere with the CRISPR-Cas system, essentially disabling the bacterial immune response. Some bacteriophages even integrate a protein into their genome that prevents the Cas proteins from cutting the viral DNA, making the virus effectively invisible to the bacterium's immune system. Viruses also evolve quickly, so CRISPR immunity may not always be effective if the virus changes its genetic code. This is why bacteria need to constantly update their CRISPR sequences to stay ahead of new viral strains, just as our immune systems adapt to evolving viruses like the flu. Who wins, CRISPR or viruses? In the natural world, there's no definitive winner between CRISPR and viruses, both are constantly evolving. For bacteria, CRISPR provides an excellent defense against viral attacks, but it is not foolproof. The viruses continue to evolve and counteract the bacteria's defenses, creating a dynamic and ongoing arms race. In the context of human technology, CRISPR has proven to be a much more powerful tool than a virus. CRISPR gene editing technology has revolutionized the way scientists approach genetics, allowing them to make precise changes to DNA in organisms as diverse as bacteria, plants, and animals. In this battle, humans, armed with CRISPR, have the upper hand. CRISPR allows us to target and edit viral genomes, effectively fighting viruses at a genetic level. For example, scientists have used CRISPR to edit the genomes of human cells to make them resistant to certain viral infections. One groundbreaking study demonstrated how CRISPR-Cas9 could be used to cut the DNA of the HIV virus in human cells, essentially disabling the virus's ability to replicate. Other research has focused on using CRISPR to target viral DNA in infected cells, offering hope for future treatments for viral diseases like herpes, hepatitis, and HPV. In this sense, CRISPR may be the ultimate weapon against viruses in the future. CRISPR-based antiviral therapies could enable the development of treatments that target and eliminate viruses at the genetic level, offering the possibility of permanent cures for chronic viral infections. This is especially promising for diseases that currently have no cure, like HIV or hepatitis C. CRISPR in medicine, harnessing its power to fight viruses in medicine. CRISPR's potential to combat viruses goes beyond simply editing the genome of infected individuals. One of the most exciting possibilities lies in its application for gene therapy. CRISPR could be used to edit the genes of patients who are at high risk for viral infections, preventing infection before it happens. For example, scientists have already demonstrated that CRISPR can be used to edit immune cells, making them resistant to viruses like HIV. This approach, known as CRISPR-Cas9 gene editing, involves modifying specific genes in the patient's immune cells so that they are no longer susceptible to infection. It's still an area of active research, but the early results are promising, suggesting that CRISPR might one day offer us a way to not just treat viral infections but prevent them entirely. Additionally, CRISPR could be used to develop new antiviral drugs that directly target and neutralize viral DNA or RNA. Rather than simply attacking the virus with broad-spectrum antivirals, CRISPR-based therapies would allow us to directly target the virus's genetic material, cutting it out of the equation at a very precise level. When it comes to the natural world, viruses and CRISPR are in a constant state of evolutionary warfare. Viruses evolve to evade detection and infection, while CRISPR provides a powerful defense mechanism for bacteria. But in the realm of human technology, CRISPR is quickly becoming a tool that could turn the tide in the fight against viruses. The real winner in this battle, however, could be humanity itself. With CRISPR, we now have the ability to fight viruses at a genetic level, offering new ways to prevent, treat, and perhaps even cure some of the deadliest viral infections. While viruses continue to evolve and present challenges, CRISPR technology is rapidly evolving as our most powerful weapon against them, and it may very well be the key to unlocking a future where viral diseases are no longer a threat. In this battle, CRISPR doesn't just fight back against viruses, it might just win.